Hello, my name is Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM, and it's my pleasure to interview today Dr. Peter Lobel. Hi, Peter.
Hi, Marty.
You want to begin to tell us about yourself?
I'm Peter Lobel. I'm a member of the CABM, resident member since December 1988, and I'm a professor at Robert Wood Johnson Medical School, currently in the Department of Biochemistry and Molecular Biology. I serve different roles at Rutgers, including being executive director of the Biological Mass Spectrometry facility, a proteomics facility.
Great. Well, Peter, you've had a very long and distinguished career as a professor and as a scientist. How did it all start? Can you tell us a little about your education and your early career?
It's been a series of happy accidents. I'm a native New Yorker, born in Yonkers and raised in Westchester County. Did my undergraduate degree at the State University of New York, and then I did my graduate studies at Columbia University in New York City, working with Arthur Carlin, doing protein biochemistry, but I also took a course with Eric Holtzman in cell biology. And Eric was a brilliant teacher and was really interested in lysosomes. And when it turned out time for me to finally leave New York, I actually was fortunate enough to do a postdoctoral fellowship with Stuart Kornfeld in St. Louis. And Stuart is one of the world's experts on lysosome biology, as well as being a terrific physician scientist. And in Stuart's laboratory, I studied molecular cell biology, really. And Stuart was working on lysosomal enzymes and how they traffic, how they get to the lysosome. And this was a really, really hot topic.
So, Peter, you finished your postdoc in 1988, and you came to CABM, so you've been here for 32 years. What was the big attraction? What brought you here?
Basically, it was the Institute and Aaron. Aaron was just a terrific guy, but we were sold on this idea that we would be able to do science and really pursue scientific excellence or to the best of our ability, and that basically if we didn't succeed, we'd only have ourselves to blame.
How would you describe the main focus of your lab right now?
Right now, the main focus of my laboratory is still on lysosomes. In the course of our studies on lysosomes, we discovered the cause of two human neurodegenerative diseases. It was a disease where children at about age 3 showed symptoms, seizures, and they would undergo progressive decline, losing their ability to speak, to go blind, become bedridden. And it's called late infantile neuronal ceroid lipofuscinosis, which is a mouthful, or also called late infantile Batten disease. And it's a disease due to the deficiency of a lysosomal enzyme called tripeptidyl peptidase 1, a protease. We discovered the molecular basis for the disease. It's a hereditary disease. So that launched a whole research program, which I won't go on all the details, but it actually led to the development of an FDA-approved therapy for this disease.
By identifying the cause, the mutation in the protein, you were able to come up with a new therapy for the disease. And is that therapy being used now?
What happens is every 2 weeks, children with this disease who are being treated receive an infusion of recombinant protein. They actually drill a hole in the children's skull and implant a device so that they can access the ventricles of the brain and administer recombinant enzyme every 2 weeks into the ventricles of the brain, and that diffuses out into the cells of the brain and replaces the missing enzyme, gets up by the cells.
So, Peter, that's kind of a dream. you work on a gene, and sometime later you have a therapy that's helping kids. So what new project are you working on now that has really grabbed your attention?
The two projects that we're working on are really extensions of our ongoing research. So one of them is to find more effective and better therapies for this disease, for this late infantile neuronal ceroid lipofuscinosis, or LINCL. And that entails figuring out ways to deliver this protein by, by intravenous infusion and having it targeted across the blood-brain barrier. The other thing we're working on is sort of the discovery of these diseases really grew out of a long-term project to identify the proteins that live in the lysosome, and which is now extended to try and identify, to catalog all the proteins, where all the proteins in a eukaryotic cell reside.
How do you interact with students and trainees in your lab?
Well, it's evolved. I used to be really hands-on and, being at the bench, and for my first 10 years, uh, uh, looking at all their data very carefully, and now I tend to be a little bit more hands-off and guiding from afar, allowing people to make more of their own mistakes, and sometimes they're not mistakes even. But
so I want to ask you one more question, Peter, and that is that I know that you are really interested in skiing, and can you tell me how science has helped you be a better skier, and how skiing has helped you become a better scientist?
the most wonderful thing about skiing is getting to a totally untracked area and going down in fresh powder and exploring new terrain and the exhilaration with that. And that's how I feel about science, that when we really focused on these little details, and in skiing you have to be focused on the little details too, so you don't hit a tree. But in science, you're focused on these little details, but the hope is you're going to open a door and it's going to be virgin territory. And then you can just track it up yourself without worrying about all the other footprints or all the other, that's to me the exhilaration and the rush of it. Right. And they have that same component.
But I love to hike, and I've had some of my best scientific thoughts on the trail, just free thinking, looking at trees, looking at the mountainsides, just thinking about how nature is organized. And I think it's really, in addition to being refreshing, it's really helped my thinking.
Well, actually, that happens to me on the chairlift.
Yeah.
What I like about skiing is you have to be totally in the moment and focused. So it's on the chairlift after doing that I have these thoughts, or in the shower afterwards. But when I'm skiing, what I love about it is I tune everything out. Mm-hmm. And you're just in the moment.
That's not too bad for a scientist also. So great, Peter. With that, we will close. And I want to thank you very much for this interview. And I hope people out in the world will be interested in the stories you're telling us. Thank you very much.
Thank you.